put on record. So as usual, I extended 7 and 8 for tomorrow. If you guys haven't done it already, please do it. I will have quiz 9 out by tonight. And if you haven't already uploaded your presentations, please upload them because I need to give you guys a grade for that. And it doesn't let me put a grade in unless you do that. So please do that. Okay, so let's go with presentations. I think we only have 2 people or today. Who wants to go first?
I don't mind presenting.
Huh?
I believe I'm presenting this weekend. I mean this week, sorry.
Yep, okay.
All right. I can't share.
Oh, whoops, my bad. Okay, you should be able to now.
Uh, it's not. It's giving me different options.
What do you mean?
As in, okay, share screen, say desktop one, iPhone, iPad, iPhone, iPad, Safari, Mail, or Universal Access.
Um, does it not show you your PowerPoint
Here,
or?
let me just click share. No, it's not. It says allow Zoom to. Can you see my screen?
Mm-mm.
Uh, hmm. Okay, let me try one more time. Can you guys see?
Yes.
I don't know what this means, but I'm gonna say yeah. Okay. You guys can see?
Mm-hmm.
Okay. So my question was question one. Let me see. Okay. And my question is, what is the initiator triplet in both prokaryotes and eukaryotes? What amino acid is recruited by this triplet? And some of the potential answers are UAA, no amino acid called in, UAA or UGA arginine, AUG arginine, AUG methionine, UAA methionine. So here's some background information to my question. Transcription and translation works together to form proteins. During transcription, DNA serves as a template for complementary base pairing, in which transfers information to a messenger RNA. Lastly, the information is translated into a protein. And each group of 3 bases in a messenger RNA is considered a codon. And each codon specifies a particular amino acid. The order of the amino acid determines what protein is going to be produced. So here is my image. You actually used this image last week. This image shows the codons and which codon produces which particular protein. So based on my background information and concluded the correct answer is D, AUG methionine, because AUG is the start codon for the protein methionine. That's it.
Good. Any questions on this? Good. Okay. Who wants to go next? Do we even have the full thing for today? Hmm. Are we missing a couple of
I can go
people?
next.
Oh, okay.
Hold on. Let me figure out how to do this. Is it sharing? No.
Yes.
Yes.
Wait. Which screen am I sharing? Is it the...
Our protein structures.
And function determined.
Okay, hold on. Wait, that's not how I do it. Ah! Hold on. I feel like a boomer. I don't know how to do this.
Present.
Go to view.
Or view.
View.
Present.
Okay, Control F5. There we go. Okay, question. How are protein structure and function determined? Answer. Protein function is determined by its shape of its primary structure, and the primary structure means the sequence of amino acids, and the sequence of amino acids is determined by the sequence of codons, which are 3 nucleotides long. This is the essential amino acid chart right here for life, essential for life. This is a polypeptide chain, which is going to eventually form into a protein. It may actually be a protein, I don't know. But yep, this shows the amino acids forming a chain, and it shows how the protein folds and fits a specific shape for its specific function. And that's all.
Good. Good work.
Oh, I stopped sharing. Oh, what am I doing?
Nimi,
Yes.
can we go over the questions we got wrong for the last exam? Because some of them I don't understand why I got wrong. Maybe you can explain to me.
Which questions do you know?
Do you mind if I share my screen?
Sure. You can do that. Are we sharing? Yes or no? No, okay.
Can you see?
Yes. Okay. In mice, presence of AA alleles give rise to agouti fur color. AA to the Y gives you yellow. A to the Y is lethal, causing mice to die before birth. The B allele results in pigment formation and is dominant over the B allele. Recessive BB genotype results in no pigment formation, gives an albino fur color, irrespectively of the presence of AA or AAY alleles. In the following cross between two mice, yellow mice, what would be the expected proportion of yellow fur mice among the live offspring? Okay. So what do we have here? Okay. Okay. Okay, this is not the easiest to show by talking. So how about you send me the questions that you have questions on, like the number of the question, and then I will turn that into a PowerPoint and then we'll talk about that next week. That way it's more visual than me talking about it. Does that sound good?
So should I like just write the numbers and email it to you?
You can either do that or screenshot it. It's up to you.
All right, perfect. Thank you.
Because it would be 10 times easier to explain these kind of questions when I actually have it set up. Okay, I'm gonna stop your sharing. That way we can continue on with what we have for today, because it's quite a bit of information. Wait.
What is this?
Ariana, are you still presenting today?
Yeah, I could present.
Okay. I don't see Carly. Okay.
Can you guys see? I don't know. Yeah. Okay, so I had question 3. Question was, how is it demonstrated that the genetic code consists of triplet nucleotides? A is RNA always contains a number of nucleotides that is a multiple of 3. B is all the answers are correct. C is an insertion or deletion of 3 nucleotides, but not 1 or 2 nucleotides within a codon region of a gene preserves the function of the resulting protein. D is an RNA that is 300 nucleotides long, always codes for 100 different amino acids. And E is every codon codes for 3 different amino acids, one for each nucleotide comprising the codon. So background information. A triplet is 3 nucleotides that are necessary to code for 1 amino acid. Background information on the triplet binding assay. Nuremberg and Letter are the scientists that were able to identify 61 different combinations of codons out of the 64 codons to the amino acids. They also discovered that the cell decoded mRNA by reading their nucleotides in groups of 3, which are called codons. And the rules for translating the language of nucleic acids to that of proteins is called genetic codes. The experiments that tested the effects of the frameshift mutation showed that the deletion of addition of 1 or 2 nucleotides caused a loss of function, whereas a deletion of 3 nucleotides allowed for the retention of considerable functions. So based off of this, I found the answer to be C, an insertion or deletion of nucleotides with a codon region of a gene preserve the function of the resulting protein.
Yep. Does that make sense to everyone?
Yes.
Cool. Okay. Okay, Carly's not in, so we'll just move on. Okay. Technology does not like me today. Okay, so this is a question with genetic code and how fancy it is. The mRNA formed from the repeating tetranucleotide UUAC incorporates only 3 amino acids, but the use of UAUC incorporates 4 amino acids. Why does this occur? Any ideas? Okay. So when you have a repeating sequence of UUAC, so we have this guy, right? So when we have this, you have it as, so you have UUA, which ends up with leucine, UAC, which is tyrosine, ACU, which is threonine, and CUU that ends up with leucine again, right? So one of the thing, okay, so, and then with this guy, when you have a tetranucleotide sequence, you have UAU, which is tyrosine, AUC, which is isoleucine, UCU, which is serine, CUA, which is leucine. So the difference between these two is that this genetic code itself is degenerate in the sense that it has codons that code for, it has multiple codons that code for the same amino acid. So in this case, the degeneracy of this code is apparent because it has two codons coding for leucine, right? Whereas in this case, it's not revealed because you have all the codons producing a new amino acid. Does that make sense? Okay. So next we're gonna talk, we're gonna start on transcription. So with transcription, you have prokaryotic, but before that, we're gonna talk about prokaryotic and eukaryotic gene structure. So with prokaryotes, you have the promoter, and anything before that is upstream of this gene. So the 5' end. And then this promoter ends with the transcription start site. Then you have a RNA coding region, and then you have a terminator sequence, which ends up with the termination site, and then everything downstream of that gene, okay? Whereas with eukaryotic gene structure, you have like enhancer controls, regulators, all that fun stuff here, which we'll talk about more if we go into regulatory, transcription regulatory sequences and all that. But that's a different topic for a different day. So we have this, we have the promoter, then we have the exons and the introns and all that. So with eukaryotes, you have introns. They don't actually code for anything, but it's still there. Exons are the regions that actually code for RNA and proteins and all that fun stuff. Then you have a poly-A signal sequence, which we will get to that in a couple of minutes. And then you have a termination region. Okay, so that's the structure of the gene in prokaryotes and eukaryotes. So when you think about transcription, transcription is essentially transcribing a gene into RNA, right? So in prokaryotes and eukaryotes, there are quite some differences, but it all goes on with three major steps. The first one is initiation, the second one is elongation, and then the third one is termination. So it starts off, so to begin transcription in prokaryotes, there is something called a core polymerase, which is made up of 5 subunits, 2 alpha units, 1 beta prime, and then an omega subunit. This core polymerase is involved in initiation of this process. So with the core enzyme or the core polymerase, it attaches it on self, it's stuck there with a sigma factor. And this is called the hollow enzyme. With this hollow enzyme, it starts to recognize specific sequences that you need for transcription. And once it recognizes that sequence of the promoter, the sigma is like, okay, core enzyme, you can go on doing what you need to do, and pretty much the sigma factor just goes on its way. Then eventually it goes on, it kind of goes into transcribing that DNA, and it's like, okay, so it sees it's like a termination sequence, and it stops there, and it's like, okay, that's pretty much how you would make RNA out of prokaryotic DNA, right? Does that make sense so far?
Yeah, kind of.
Okay. So that's with prokaryotic transcription. So when you see this guy, so we have a prokaryotic gene. So we have this, it's prokaryotic, it's DNA. When you transcribe it, you end up with a UTR here, 5' UTR. You have your RNA segment of what was transcribed from here. And then you have something called a 3' UTR that is there, and it's like it has the stop codon here, termination site ends up coming there. Does that make sense? So the polymerase is like so the polymerase is here. It's like code, terminator, stop, stops what it's doing, and then it has this area.
So like the whole blue DNA part becomes the promoter.
Wait, say that again?
Can you go over like that again? Because I didn't get it. How like the DNA, the whole DNA blue part of it became like the green promoter part.
Okay, so this is not what is being coded. So in this case, so if we go back here, the hollow enzyme here is going into the so the hollow enzyme is with the core enzyme and the sigma. So this guy is just going through until it finds that promoter. The promoter is not transcribed into the RNA. So that start site, so we have that promoter. So the core enzyme comes here, and it's like, okay, the sigma factor here falls off, and it goes through this sequence, adding on the complementary nucleotide from for RNA, okay? So that happens, and then you have this region getting formed. And then these are like caps to the mRNA. Does that make sense? Okay. So next, we're going to look into eukaryotic transcription. So with eukaryotic transcription, you have so to initiate this, you have a Tata box. Tata box is a region in the DNA itself. It's typically around the promoter area. And this Tata box brings in something called a Tata box binding protein and transcription factors that are involved. So in this case, this is called transcription factor 2D, okay? So that guy comes in and it's like sitting on the data box. And it's like, okay, we need to get moving here, people. And it's like, so these activators and transcription factors, they pull in the type of RNA polymerase that it needs. So with eukaryotes, we have, or in general, we have 3 types of RNA polymerases. The first one is for ribosomal RNA. And then the second one, we use it for messenger RNA. And then the third one we have for transfer RNA, okay? So in this case, we have, so the transcription factor 2D pulls in 2B, so transcription factor 2B, RNA polymerase, and transcription factor 2F. So those all come in and they're like, okay, we got to pull in our other buddies called transcription factor 2E and 2H. When those two all come in, the RNA polymerase 2 is like, yeah, okay, we got this, right? And then they just like continues on until it sees a region where it's like terminating, right? So they see this kind of region there and they're like, okay, we're done. And then it sees a polyadenylation set. So that's just a sequence to add on a poly A tail. So many adenines adding on. Does that make sense? Oh. Hold on. Are we okay? Somewhat?
Meh.
I'm lost in the sauce.
Me too.
Oh boy. Okay.
If I'm being honest, that's
Same.
usually.
Okay. So let's see. Okay. Let me see. Oh no. Okay. So, okay. Let's see. So, initiation. What is happening here? We have, oh wait. Okay. We need our brains working. I know. Okay. So, we have our data box, right? It's a sequence in the DNA. This is bringing in other components that allow you to activate. So, okay, so we have, so for this to occur, you need the different transcription factors, right? So these transcription factors allow it to propagate more. So these guys come in, they're like, okay, so this guy here is telling Chatterbox, it's time to get the others ready for work. So this guy is going like, okay, guys, come on. And then it's like, this guy binds. When it binds, it's like pulling in its other friends to help out. So it's like, okay, so B and F and the polymerase comes in, but the polymerase is feeling like it's unmotivated. So they bring more of the friends or the transcription factors here. They're like, come on, guys, we need to motivate the polymerase to actually do its job. So these guys come in, they're like, okay. Then they kind of get the RNA polymerase to get started, get those signals running, and then RNA polymerase 2 just zooms off transcribing. So adding on the different nucleotides you need. And then once the RNA polymerase sees this region, it stops. And then it adds on a poly A tail. Does that make sense? Or did I lose you even more?
I'm still lost.
Okay, where am I losing you guys?
Okay, what do the 2B, the 2F, the 2D, the 2E, the 2H stand for again?
So those guys are transcription factors.
Transcription factors. Okay.
Mhm. So
So
those guys, oh,
no, go ahead.
so those guys are needed for this process to start. So it's part of the initiation complex.
So it doesn't start until all 5 of them are together,
Mhm.
and then it starts.
All of it.
Mhm.
And this is only messenger RNA, right?
When you're talking about RNA polymerase 2.
Okay. RNA polymerase 2.
Mhm. So with transcription here, right now, we're only talking about messenger RNAs because that's what leads into coding proteins.
Okay.
Any other questions? Concerns?
I think I'm good for now.
Okay. Okay. Trust me, we will spend another week on this too. So if we have more questions, we'll go through them. I just want to make sure I have at least the basics down for you guys. That way you can actually do the mastering genetics assignment. Okay, so again, so now we have our pre-mRNA going on, right? We have this pre-mRNA, and we have all the exons, introns, all that fun stuff. Now, what do? We need to get rid of these introns, right? And then protect the ends of our pre-mRNA. So to do that, we go through a process called RNA processing. This involves 3 groups, which is the 5' capping, the 3' poly A tail, and splicing. So in prokaryotes, this is not necessary because you don't have any introns, so you don't have to process the RNA before going into translation. It just goes directly into translation. But with eukaryotes, you see this with the with eukaryotes, you have this situation where you have these introns, and you're like, oh, we need to modify it before it goes into the cytoplasm because it needs some protection from any other components that are involved while it's being transported out, right? So in this case, we have our pre-mRNA. That guy comes out, we have an exon, intron 1, exon 2, intron 2, exon 3. We have 2 introns going on. We're like, okay, we're gonna cap it with a methyl 7 guanosine group at the 5' cap. So that's what's gonna go there. And then the 3' we have a poly A tail. So many A's just added to the end. And then they take out all the introns. Does that make sense so far?
Kind of, yes.
Okay. Oh, okay. In which cellular organelle do the 3 post-transcriptional modifications are often seen? In the maturation of mRNA in eukaryotes occur. So where do you see
Nucleus.
RNA? Oh,
The nucleus.
nucleus, yes. So that's where that occurs. If it asked for prokaryotes, What would you say?
Is it a cytoplasm?
So, oh, did somebody else want to say something? Okay. So remember, guys, this post-transcriptional modification only occurs in eukaryotes. So none of these answers would be right. Right? Does that make sense?
Yeah.
Okay. So then there's something called alternative splicing. This is a way to get different isoforms of proteins. So it depends, sometimes it skips over a specific exon. It's like, okay, we need red, yellow, and blue protein here. So they're like, so the promoter's like, guys, we're gonna splice out the green exon in here. Okay, so when this guy goes away, you get this. So it splices it out. It's like red, yellow, blue. It comes down and it makes that protein. This here comes down here, and then so this guy's like, okay, we need red, green, and blue. So they splice out the yellow and give that spliced mRNA, and then you end up with that protein. So that's kind of the idea of alternative splicing. Does that make sense? No?
Just what is the last step? Like when they make random steps? Like
Wait, say that
shapes.
again?
What is like the protein isoforms? How does it do that?
Well, we're gonna talk about translation next, so we'll get to which protein it is, but the splicing part, it just depends. So the splicing itself depends on what the promoter is telling that gene to do. So depending on what signal that promoter gets, that's how you end up with which splicing set you have. Does that make sense?
You can only splice with a promoter.
So the promoter is what's telling the gene what it needs to do. So it's, the promoter is like the boss. The boss is telling everyone else, okay, so we need, so whatever signal is coming into the promoter, it's telling, guys, we need the red, green, and blue protein, right? So that way, that promoter is like saying that, and then it transcribes it out and then has it splice out in that manner. Does that make sense? Okay. Any other questions before we go into translation? Okay. So translation. Translation is the process of converting the information in an mRNA molecule into a sequence of amino acids. And these end up making up proteins, right? So translation requires ribosomes, transfer RNAs, amino acids, and energy. So in this case, we need ribosomes, right? What do these ribosomes do? Ribosomes actually provide the scaffolding on which the tRNA and mRNA are brought together with some enzymatic activity to actually form the peptide bonds, right? So in this case, with prokaryotes, you have a 70S ribosome. And it's made up of two components called 30S subunit, which is the small subunit, and 50S subunit, which is the large subunit. Okay, so these guys are there, and you have many ribosomal RNAs going on. Whereas with eukaryotic ribosomes, it's called the 80S ribosome. It has a 40S small subunit, 60S large subunit. Okay? So that's with ribosomes. With tRNA and all that fun stuff, so these guys have a specific amino acid that is covalently attached to this 3' end of the tRNA. And this ends up binding the mRNA at that codon, right? So it has the complementary sequence to that codon to decode that code that is in the mRNA. So tRNAs are typically in a cloverleaf shape. It's a secondary structure for RNA. And this loop here is called the anticodon loop. And it has what is defining what codon it is decoding. So it's the complementary sequence for it. So example, 5' CCA pairs with 5' UGG, which ends up with a tryptophan up here. Does that make sense? Okay. So next we're going to go into the steps of translation. So the first step is initiation. With initiation in prokaryotes, you have an initiation complex, which involves the small ribosomal subunit, some initiation factors, and mRNA at the AUG subunit, okay? So when this happens, and then that complex itself comes in and combines with that little, the large ribosomal subunit. This ends up, so this unit here ends up going to see this sequence called the Shine-Dalgarno sequence. It's this sequence here. It's a little bit ahead of the start codon in bacteria, and it's complementary to the 16S rRNA sequence here. That way it can come and bind and start off the process of translation in prokaryotes. Whereas with eukaryotes, you have the recognition sequence here at the, so the 5' cap here also serves as a recognition sequence. So once they see that, they're like, okay, they go through that RNA and they're like, okay, so where is that initiator codon? And then they are like, okay, so once it's there, it starts. That's how it's initiated. Does that make sense? Okay. Okay, so initiator codon bringing in methionine, and then off it goes. So when it goes through, so the next step here is elongation. When it's elongation time, so the tRNAs bring amino acids one by one to the polypeptide chain. And in the ribosome itself, there are 3 sites. The first one is called the amino acyl site. This binds the incoming charged amino acyl tRNAs. So this tRNA has that amino acid bind to it. And they come in, they're like, okay, let's do this, right? Then it goes into the peptidyl site. When you hear peptidyl site, think peptide bonds, because that's what they're doing. They're essentially forming peptide bonds right there, right? So that's what they're doing there, but they haven't fully dissociated it from the tRNA itself. Then you have the exit site, which is the E site. The E site ends up releasing the dissociated tRNA so that they can end up being recharged with amino acids, right? So So that's what happens with elongation. It's just a repeating cycle until they reach the stop codons, right? So termination occurs when the ribosome ends up reaching a stop codon. When this stop codon occurs, there is no complementary tRNA that goes with it. So in this case, you end up with a release factor coming in, binding to that stop codon. It stops the ribosome, releases it, and frees the tRNA and the polypeptide chain. Does that make sense? Okay. So one more slide, I promise. I know this is like information overload now, but just a little quick overview on protein structure. So the protein structure itself depends on the sequence of the amino acids. And then when it comes into like the secondary, tertiary structures, the structure determines what happens with the function. So with this, it's like, so the primary structure you have here involves the sequence itself. Then you have it looping up or going all sheet-like when it's in their secondary structure. Then the tertiary structure ends up with multiple of these guys just looping around, kind of trying to get their own form, like a 3D shape. And then the quaternary structure is just multiple protein molecules and multiple polypeptide chains coming in together and being like, let's do this. Okay, so that's all I got for today. Do you guys have any questions, concerns? Did I freak you out more than I should be?
No, my brain just hurts now.
Yeah, it was a lot.
I know, I know. I just wanted to make sure you guys had enough information for the homework itself. But any questions?
That was so much.
I know, I know. Forgive me. I'm debating whether I should just count attendance as the quiz, considering the amount of material we went over. What do you guys think?
I wouldn't mind that.
Wait,
My brain's like fried right now.
I wouldn't mind that either.
Okay.
If I learned this at the beginning of my cell bio class, that would have made things so much easier.
Yeah.